Amin Arbabian received his PhD degree in EECS from UC Berkeley in 2011, and in 2012 joined the Stanford University as an assistant professor of electrical engineering, where he is also a School of Engineering Frederick Terman Fellow. His research interests are in high-frequency circuits and systems, medical imaging and screening technologies, and ultra-low power sensors and implantable devices. Professor Arbabian currently serves as an associate editor of the new IEEE Journal of Electromagnetics, RF and Microwave in Medicine and Biology, and also he serves on the technical program committees of RFIC and ESER conferences. He is the recipient or co-recipient of the 2016 Stanford University Tau Beta P Award for Excellence in Undergraduate Teaching, 2015 NSF Career Award, 2014 DARPA Young Faculty Award, and Best Paper Awards from several conferences, including ISSCC in 2010, VLSI Circuits in 2014, RFIC Symposium twice in 2008 and 2011, and several other conferences. With that, please join me in welcoming Professor Arbabian.
Thank you. Thanks, Mujtaba. So thank you, Mujtaba, for the organization and also the Solid State Circuit Society for organizing the talk, TI for hosting it, such a wonderful venue, and we are very close to, yes. So my talk is basically going to be about relationship between, if there is a relationship between Nyquist rate and healthcare, and the idea is how do we design electronic systems to close this sampling gap. There are two takeaway points I hope that you have, right, based on this talk. The first one being that there is such a crisis in healthcare, that we do have a crisis, and the word crisis, I understand these days, you turn on the news, there's lots of crises, but if you still care, there is a crisis in healthcare. And the second point I wanted to make is that technology and electronics in our society has a role to play in solving or improving, mitigating this crisis. So those are the 2 points. Now, before I get started, let me introduce my group. So we work on, at Stanford, we work on innovations in RF systems. So my group has 3 different parts. The first part of my group looks at millimeter wave systems, imaging, communication devices. We answer questions like, how do you do fiber-like speeds over wireless channels? How do you design, so we have a slight wireless problem here, but so one of these problems we're looking at is how do you do longer range wireless systems, point-to-point system, designing a lot of chips. Second part of my group is related to imaging systems. How do you design multiphysics, hybrid imaging technologies that sort of close this gap? I'm going to talk about this a little bit today. There's a little bit of work on biosensing, and finally I have a work on ultra-low power sensors for a variety of applications, including IoE and implantable devices. So incidentally, last time I was here, my talk was on IoE devices. We looked at low power devices, low power sensors. So today I'm going to talk a little bit about healthcare. It's a little bit different topic. So let's dive into it. The outline, I'm going to start from discussing cost trajectories as big crisis. I'll talk also about poor outcomes for a variety of disease states. So these are the fundamental problems of healthcare today. Then I'll talk a little bit about what Stanford Medical School sort of calls precision health versus precision medicine, what their vision is and how technology can fit into it potentially. And finally, I'll talk about new technologies and some of the work we're doing in my group sort of to address some of these problems. There's 3 projects I'm going to talk about, and they're going to start from the outer of the body, start from monitoring devices, going inside and all the way to the implantable devices. So non-invasive completely all the way to sort of implantable devices. So let's dive in. Okay, so healthcare costs. So the first slide is, I'm sure you've seen many, many times. The idea here is that if you plot healthcare expenditures as a percentage of GDP, it's growing at a very fast rate, and periodically you get these flat regions. This is where your economy is growing at a faster rate, your GDP is going up. Then you hit sort of a recession and something like this happens. So if you plot it after 2010, you see that it's flattened out a little bit. But most of the issue is that the trajectory is always positive and it's not sustainable. If you look at it, it's going to be about 30% of GDP by 2030. That's twice education and defense combined. If there's anything left in education by 2030, that's going to be included in this sum, I guess. Aging population aggravates the problem. And the question we're trying to answer is, usually when any time cost is an issue, our instinct in our society is that applying technology will bring down the cost. The question is, does it apply to healthcare? That applying the technology will bring down the cost. Okay, so if you look at the breakdown, so we got to figure out this range problem, I guess, a little bit. So if you figure out the breakdown, you see that if I plot the cumulative distribution of the healthcare cost, about 1% of spenders are taking about 20% of the system cost. About 5% are almost half the entire cost. So what this tells you is that you need to look at it from a very specific point of view. For example, early detection becomes very important because the healthcare cost rates are essentially going to get affected by complications. The more complications you get into, the more overall cost goes up. Second problem is chronic disease management. How are you going to attack long-term chronic disease? And third is how do you go from this one-size-fits-all systems to more personalized care? So these are the sort of high-level intuitions behind how you can potentially bring down the cost curves from a technology perspective. Now, I was very curious when I looked at these cost curves and I said, well, where are these costs going? What are the potential areas? And I bring a couple of examples here. The first one is diabetes. So if you plot the cost expenditure related to diabetes and its complications, you see that in North America, we have $350 billion today spent on diabetes and complications. Now, if you put it in perspective, the semiconductor industry as a whole is about a $300 billion industry, and we have diabetes as $350 billion today. This doesn't even take into account growth that we have in this space and also other related disease. A second example, which was interesting, is Alzheimer's. So here we see that today we're about $300-some billion, and so we're about 4 or 5 million patients, but it's growing at alarming rates, and so something has to be. So these are where the costs are going. If you're curious on what adds up to a few $1 trillion, this is where the costs are going, and trajectories are not positive. Now, cost aside, you might argue, well, we're spending a lot of money, but at least we're getting a lot of good health care. The quality must be good. The second part of my argument is that the quality is also not as we expected it given the cost. So here I'm plotting mortality rate from heart disease and cancer from 1980s to about 2010. And what you notice here is that we have done a much better job in bringing down the mortality rate from heart disease. And as far as cancer goes, we're pretty much flat. If you see a downtick in sort of 1990s, some of this is associated with changing in smoking habits. So as a whole, we haven't done much in cancer mortality rate. And this is in the era of introducing all sorts of technologies and screening technologies, medical imaging technologies. We have MRI, PET scans, CTs, combination of MR, CT, PET scans, more advanced markers, fMRIs. We have everything at our disposal, but at the end of the day, this is remaining flat. So that's another question to ask, is how do you sort of get quality out of the healthcare system? So let me give you one take on what the problem may be in terms of quality. And the take I wanted to discuss is what we see as a Nyquist problem. Here, I'm plotting sort of conceptually the cancer progression rate on the y-axis and time on x-axis. And so the idea is we have different types of tumors, and we have sort of a sampling of screening intervals. And these screening intervals are set essentially by cost or by safety. You don't want to go through a mammography system or x-ray system on a daily basis, on a weekly basis. Even if you wanted to do that, there'll be a cost associated. So what dictates these sampling intervals today are a combination, depending on where in the world you are, cost and safety. So this is set by about 18 months. So somewhere between 18 to 24 months. Now the problem with this is if you look at the most aggressive type of tumors, the ones that we're most concerned about, And this sampling rate is not fast enough. And most of the time, patients are going to the hospital, to their doctor, saying that they're feeling something, there's a problem, and the diagnostic test is being run, as opposed to screening showing up something. So there's a lot of what we call interval cancers, where something happens between the intervals. And so what we are catching is a lot of these slow-growing cancers, and this is not bringing the mortality rate down. There's lots of different studies on this thing, and one of the ones that is more relevant is looking at graphs like this, where you look at, from 1970s to 2007, you look at the rate in different types of, finding different types of cancers, localized, regional, and distant. Now, if you introduce mammography, you see that there's a huge uptick in finding localized cancers. So that's great. So you're definitely finding more cancers. But the problem is the ones we're finding, there is no distinction. We haven't tracked them. We don't know how they're growing. We've just taken one sample and we've identified there's a problem. We don't have multiple samples. We don't know what type of cancer. So most likely these are falling on the slow-growing tumor type C and D. So, but if you look at regional or distant, it's pretty much flat. So we haven't changed what we really need to change. And the studies on this thing are numerous, but the ones I want to bring here this is the 13-year study of about 40,000 women in Canada that looked at mammography and kind of concluded that it's not associated with the reduction in breast cancer death. And there's similar studies, if you follow the news, there's changing guidelines on a sort of yearly basis. And there's been a similar thing that just came out on a different type of a screening system a few weeks ago. So the next question we want to ask is, if you look at healthcare today, the system of healthcare today, you pretty much have combination of two things. You have advanced diagnostics, which means that better imaging tools, better diagnostic tools, more advanced, more expensive diagnostic tools. And then at the same time, we have precision medicine, which is once we figure out something's wrong, we go in with all the tools and technologies that we have at our disposal, try to fix it. These are the state of the art. This is the direction of research. This is the direction of funding and everything else. And the problem with this is we don't even know oftentimes how to define health and disease. When we take a sample, when something's wrong with you, you go to the hospital, you take an MR scan. 5 different things are going to show up. Where is the problem? Because we've only taken one sample, it's oftentimes difficult to associate that sample with what has gone wrong. We don't know what your baseline is. Are you supposed to have a heart rate that's beating this fast? Are you supposed to have a high blood pressure? Or is this a normal thing, or is it not? So baselines are very important. We don't know what the health and disease state looks like. So this is what we have today, few samples. You might be going to the hospital or your doctor once a year, measures your heart rate, measures your blood pressure, measures your weight and height, and maybe once in a few years you do a blood test, and that's it. These are the number of samples we're taking. So when something happens, we don't know where this is coming from, and we don't have any way of quantifying the parameters. So what we have here is, this is what we call precision medicine. The goal is, if you want to get to precision health, you need to take much more samples in terms of quality, in terms of quantity. You need to take more frequent samples, and you need to do that at a much higher quality than we do today. And so our goal in precision health, I put it in quotation mark because this is what Stanford is looking at, medical school, is proactive, predictive, and preventive care. And so you look at the problems with much higher resolution in terms of the parameters you're measuring and how often you're measuring these parameters. And in order to do this from a technology point of view, we need low-cost, easy-to-use, and accurate screening, monitoring, and therapy tools. We need to look at it from a slightly different point of view than what we do today in the current healthcare system, which is essentially based on something goes wrong and we have all the tools at our disposal to fix it. I wonder if we could maybe move the laptop a little bit higher, because this wireless range is a little bit of a problem. So if I wanted to summarize everything to sort of one slide, I would summarize in the following way. I can have the y-axis, you're looking at utility, so how advanced is the parameter I'm trying to measure, and x-axis could be frequency of testing. So today we either have these advanced diagnostic tools, MR, PET, CT, and blood analysis, and so forth, done very infrequently, or we have something that's done much more frequently, but the parameters are not as rich, not as relevant. You look at all the variables, sleep monitoring, parameters, activity monitors, and so forth, heart rate monitoring. These are not giving you the type of resolution you're interested in terms of quality. And the question we want to answer is, can you go to next generation devices that have higher frequency of test and a much higher quality? So that's the goal. This is what we call it a sort of a Nyquist problem. And does technology play a role in that realm? Now, obviously, for this to really happen, you also needed to have low cost and safe. You need these devices to have the ability to be ran on a much more frequent basis. You need to be specific and robust. And most important of all, which is some topic for a different talk, you need to sort of manage false positives. Because if you have false positives and you're doing more frequent tests, you can imagine things are not going to go well. And so I won't go into false positives, a whole different talk, but you need to really run your statistics on your false positives when you introduce a new frequent test into the system. There's a lot of things you don't want to know, essentially. Okay, so I'm going to kind of propose a few cases of technologies that we work on. And so the first case is on cancer screening. So we were looking at what are the missing links if you want to have a more effective screening tool from my personal perspective, my group's perspective. So firstly, you need to have this available and frequent. So it doesn't matter if you're doing a test that's every 5 years. That's not going to be helpful because a lot of things happen in 5 years. If you look at screening, you need to be more frequent. Second thing you want to do is you want to make sure it's accurate. So from contrast, resolution, and false positive perspective, you need it to be accurate. So that's the second problem. And ultimately, our sort of secret goal is to make sure we move it closer and closer to the patients. And now, of course, there's a lot of implications in terms of FDA regulation, in terms of putting something in patients' hands. Well, that's why I call it a secret goal, because it may happen in the next 10 years, it may never happen, we'll see. But the idea is you want to move it closer, and you don't want to have the analytics close to the patient necessarily, but you want to have the device that's measuring any parameter sort of close to the patient. So when we start looking at this problem, there's a lot of different options. The first option we looked at was, what about microwave imaging? We know how to design microwave sources for this purpose. And it's very scalable and low cost, potentially scalable and low cost. We know how to design these things, essentially. Right? Now, there's a couple of problems. I did a lot of work in this microwave imaging space. And if you're looking at low-loss tissue or close to the skin, it may work well. But for normal tissue, there's a very severe range resolution trade-off. And the range resolution trade-off is shown by these bottom graphs here. One, on the left, I'm showing the body fat. On the right is body fluid. So I'm showing you penetration depth and resolution as a function of frequency. So when you go to higher frequencies, you get better resolution because your wavelength is smaller from a radar perspective, but your penetration depth drops. So it's a trade-off, and you have to select your frequency depending on your application. So in this particular case, you'll see that if you go to body fluids, at some point, actually the resolution is less than your penetration depth, or equal to penetration depth. So that means that the imager becomes useless. I can't go deep enough to see any pixels or any kind of voxels. And the associated problem with this is that because I've used frequency as my degree of freedom, there is no additional degree of freedom to use. In other words, if I wanted to optimize for contrast, which is a big problem, I don't have any parameter. And we know that endogenous contrast for dielectrics are modest, they're not huge. So the question is, how do you actually overcome that problem? Now, the answer to it partially could be that what if we go lower in frequencies and try to do some algorithmic work to get the resolution back, work on super resolution, inverse problems. There's a lot of good work on these problems on sort of numerical electromagnetic side of the house, numerical microwave side of the house. A lot of good work by professors in Wisconsin, so Professor Susan Hagnes and so forth. But the fundamental problem there is as you go to lower frequencies, the radar cross-section of your malignancy, of your target, drops. So the SNR becomes a huge problem. And we know resolution, super resolution, all these things work well if I have SNR. Don't have SNR, that's going to be a problem. So for that reason, we started looking at multiphysics imaging. We said, okay, microwave imaging on its own has some problems. It's good for a particular set of problems, but not for every problem. How do you come up with something that's better? And we started looking at multiphysics. And the first physics that we looked at was thermoacoustics, or thermoelastic response. This, it turns out, was invented by none other than Alexander Graham Bell. And this was an invention that he thought would be more valuable or more interesting than the telephone. He called it the photophone. And the idea was if you shine a pulsed sequence of laser source or heating on an object, it will expand and give off acoustic waves. So if I shine a laser source or a focused light source on an object, it instantaneously expands on a very small scale, but this expansion leads to acoustic waves that I can pick up. So he was trying to do wireless sort of communication back then by using this idea, right? So more recently, in a modern world, this has been used in photoacoustic or thermoacoustic imaging. So you go in with a laser gives you this expansion, the ultrasonic transducers sort of pick it up. Now, why it's interesting from a technology point of view is that I understand how to go in from either a laser or microwave source, and I can understand how to design ultrasonics, so that could potentially, again, be a low-cost solution, right? So the physics of the problem is that you generate pressure by heating. And the heating is coming either by photonic excitation or a microwave excitation. Now, the problem with the photonic excitation is that the depth is going to be limited by scattering or losses. Photons don't actually get into tissue as far as you'd like. With microwave excitation, you can go actually much deeper. So that's the advantage point. Now, one of the things to keep in mind when I first looked at the equation, one of the things that struck me as most interesting was that the pressure that I generate, I don't know if this is visible, but the pressure I generate is proportional to the intensity of the RF wave that goes in. What that means is that the acoustic intensity or power that I get is proportional to the RF power squared. And normally in radar, I put power in, I get power out. I double the power, I get double the power in. So look at radar equations, it doesn't really matter what sort of power bandwidth you use, the energy per pulse is what matters at the end of the day, right? But here things are different. If I put double the power in RF, I get 4 times the signal, right? So that's one problem you have to look at when you're looking at it from a detection point of view. So you're proportional to the square of the deposited energy into tissue, and that has implications I'm going to talk about a little bit. Now, when you look at this in more details, you see that, in fact, the interactions of electromagnetics and ultrasound are not just confined to thermoacoustics. Thermoacoustic is one of these interactions where you go in with energy, that energy generates currents, J dot E generates heating, and that gives you the expansion you're picking up. It's a nonlinear phenomenon, right? But also, if you have a static magnetic field and you generate eddy currents, we get Lorentz forces, J cross B, and that generates also acoustic pressure. You can do imaging based on what we call magnetoacoustics. So today I'm not going to talk about our work on magnetoacoustics. I'm going to skip that today. But you need a static magnetic field. It doesn't have to be as accurate as an MRI system. It just has to be strong enough. It doesn't have to be homogeneous because I get the resolution from acoustics, not from the field intensity or the gradient accuracy. And ultimately, you can also do electroacoustics where you're just looking at charges or ions, and they also respond to electric fields, and they move around. And as they move around, they give off pressure waves, and I can pick that up to do imaging. So I'm not going to talk about these two. This talk is going to only focus on the first topic. So theoretically, I have microwave excitation, which gives me spectral information and contrast, and I have ultrasound, which gives me resolution and structural information. Combine the two, I get sort of best of the two worlds. This is the good side of the story, right? So I get independent depth and resolution. So I can now select my frequency to be optimal for contrast, because the acoustic frequency and the RF frequency are now independent. I can choose the RF frequency for penetration, absorption, and contrast. I can use the acoustic frequency for resolution and structural information. So theoretically, I can get both. There is no self-interference. If you've designed radar, you know that one of the big problems, GPR, different type of radar, is what we call a self-interference. So you transmit and receive, and your transmit sort of interferes with your receiver. They're sitting right next door. There's less crosstalk, and there's multiple degrees of freedom. So this is the plus side of it. The downside is if you look at thermoacoustics today, if someone wants to go in thermoacoustics today, you need a very powerful pulse source because the transduction efficiency is low. So the way it's done today is you get a magnetron or a klystron, vacuum source, something that's pretty large, as tall as I am probably oftentimes, and you pulse tissue with that microwave source. You pulse it at megawatts or hundreds of kilowatts of power. and then you listen to the acoustic signal that comes out. So this is what's been going on in thermoacoustic photoacoustics. In photoacoustics, you use the equivalent laser that has tens of millijoules, 10 to 20 millijoules of energy per pulse. So again, hundreds of kilowatts. If this pulse is a nanosecond and it has 20 millijoules, we're talking about a very large power per pulse. And so we are limited in terms of resolution, or in terms of signal-to-noise ratio. Now, you might ask, well, if you want this to be scalable, you really need to do it in semiconductors. You need to do it in 3.5s, gallium nitride, eventually maybe silicon. Doesn't work if you have vacuum sources. It kind of defeats the purpose. Now, so what do you have at your disposal? I'm limited in my peak power whenever I go to semiconductor sources. So the only thing at my disposal is to have a longer and longer pulse width. That's how I deposit more energy, right? Now, problem with this is as you increase your pulse width, just on radar, you start losing resolution. And you see the measurements we did for this. We have 200 nanosecond pulses all the way to 10 microseconds. And you see that you can see sort of that the SNR is improving, but your resolution is sort of dropping as you go higher and higher in frequency in your pulse width. So that's a major trade-off. And also at some point, the received TA signal at the specific bin actually saturates. So if I have a longer and longer pulse, at some point I don't get any benefits in SNR. So that's a fundamental problem. People have seen this problem and said, either give up on thermoacoustics or we're going to use a vacuum source, a very large, powerful source. So when we looked at this problem, we saw a lot of overlaps with radar. Radar started out as a pulse system. That's technically not true because the first radar was actually CW. But most major radar systems started out as being pulsed. Over time, they had coherent processing applied. Everything that we've learned in coherent digital communication was applied to radar and vice versa. So we said, why can't we apply the same thing? Why can't we do coherent processing, have longer pulses, but coherently process it so you get the resolution back? So look at the amplitude and phase simultaneously. There's a couple of problems here. So you improve SNR, but the problem is, unlike radar, here we're going in with microwave, which is about gigahertz, and we're coming out with ultrasound, which is megahertz. So coherently processing these two is not obvious. And the solution for this we have is we take the envelope of the microwave signal, and then we modulate the envelope. So you apply any kind of modulation you want, the chirp or the CW modulation, sort of to the envelope. And remember that I told you that the pressure is proportional to the heating, which is the electric field squared. So this phenomena gives you the demodulation for free. So essentially you have an AM demodulator that's sitting into your physics. If you go in with an envelope that's modulated, you get your acoustic signal back for free because the physics will do the demodulation for you. So we take the microwave, and that's essentially our process. So we actually designed two sets of systems. On the left, I'm showing you the SFCW system, the step frequency version, where you take a coherent source, your signal generator, you take the envelope of this and you modulate it with tones, and then once you process it, you go through the imaging, you get the same tones back at a different frequency. The envelope of these tones back at your acoustic frequency. You don't care about the microwave. Your acoustic detector only looks at the acoustic side at megahertz. You can also do this with a frequency chirp, where you take your signal and you chirp the envelope. So you take the envelope and you chirp it, and when you look at the received signal, you're going to see the demodulated version of that chirp in the acoustic range. So we're doing coherent processing across two different frequencies here. And if you do this, essentially you would allow for semiconductor-based TA imaging systems. Because now I can get a very nice trade-off between pulse width and peak power. So with a lower peak power, I can increase the width and get my SNR back, even at a lower extent, but it's possible. So these are the images what we've done. So this is an RFMD-based gallium nitride PA module. So it's a compact PA that we're designing, no longer something huge. And you can see that in a pulse system, my SNR is severely limited, even with a simple target, very simple target. This is a piece of chicken meat in an oil phantom. Simple as it gets, VW and FMCW, my SNR comes back. I get my SNR improvements. And on the bottom right, this is something we recently published, is that you can actually look at complex tissue structures now. And this is not a simple tissue structure anymore. You have a combination of muscle fat structures. And you see that this projector is not showing everything, but you will see the layers showing up. So we're getting closer. We're not there yet, but we're getting closer and closer to implementing something that can give you resolution, although with a smaller form factor, with a variable, towards a variable, let's call it, form factor. So the second problem we started looking at with same technology is, can you go through the skull and look at internal hemorrhage or hematomas? That's another area that you need a sort of on-site detection system. You also need it for stroke differentiation. You have two types of stroke, different remedies. I need to know which one, and you can do that if you can go through skull. So we started looking at measuring targets below the skull. We have a brain phantom, looking at the layers of this brain phantom. There's a lot of work to be done, but point I'm trying to make here is we are seeing the potential of this system to be able to reconstruct images that have relevant resolution and contrast to make decisions, or at least track a parameter of something that's we call active surveillance. So you surveil a particular object in tissue over time, but now you can do it on a daily basis. You don't have to do it on a yearly basis, right? So that's sort of the idea. The third project we're looking at is we're looking at the wearable. We're looking at breast cancer detection, and we're looking at the wearable device that can do that. So we actually built a multi-element array that's 3D printed, sits around the tissue, and looks at objects inside. Now, why did we design an array? The reason we designed an array is we have one trick up at our disposal, which was increasing the pulse width, doing coherent processing. That gave us 20, 30 dB in SNR improvement, depending on your pulse width. Another trick is, what if you have an array, you can coherently sum the signal. We know about beamforming. This community has been doing beamforming for a long time. Can you apply the same thing in tissue? The answer is yes. So instead of depositing that energy, the limited energy you have everywhere in tissue, you now focus it sequentially through different areas of tissue. And in every area, you have more power, more SNR. So you can get improvements in power combining. Now, there's actually a side benefit. There's a small side note of why you want to use arrays. It's a subtle point, but it's actually more important, is that if you only have one antenna and you put it next to tissue and you excite it, the hottest point, the SAR limit, the thermal regulation FDA limit, is going to be set by the point at the surface. So the deeper you go, the energy dries with exponentially with the losses in tissue. Now I have 10 antennas around, I can make the hot spot, the hottest point, be my target point instead of being the surface. That's a huge improvement. It's a subtle point, but that's actually what we win by using an array. So what you need is a high power version of phased array we design. So the peak power could be higher, so you need to design high power peak systems. So here's a simulation of multi-antenna systems, and you can see that in simulations we can move actually the hotspots around with some limited resolution depending on your frequency by changing the phase of these elements. It's not a perfect phased array because the wavelength is still large, but it's sort of a mid-field, near-field sort of phased array system, if you think about it. So we designed the systems. We have tissue phantoms, again, are simple phantoms, and you can see by moving the focus from center to the sides, I can improve my SNR here to here. If I have two targets, again, I can move my hotspots from the center to the side, and I can change my SNR, and I can get SNR improvements, essentially, but this is so. Now, the next question is, well, contrast and SNR seems to be something we can work with at least. Well, what about resolution? Can we see targets that are small enough? So we started having a whole new project looking at resolution. So here was one of the oldest studies we did on this thing about a year and a half ago. And we looked at capillaries. So these are microcapillary tubes that are 280 micron inner diameter, 640 micron outer diameter. We put saline solution inside, and we saw that can we actually image these things with this system? Do we have that sort of an ultimate resolution in sort of a simplified setting like this? And you see that, yes, we can, to some extent, we can get the signal reconstruction back. So we took that and said, what about if we make it a more complicated sort of trade-off between signal level and resolution? And you can see on the right, for the most of these experiments, we're actually getting the resolution and reconstruction of these small capillaries, and at some point, the noise interference from the sides actually dominates. At this point, we're getting limited resolution. But in these three other cases, we're able to sort of reconstruct these 600 micron capillaries inside. So the resolution ultimately is there. It's a matter of optimizing for it. And then our next experiment was, well, all of this is good in capillaries, but does it work on real tissue? We haven't done this on in vivo yet, but we looked at, or the first step, We looked at a scallion plant. So we looked at the vessels in a scallion plant. You can see here, this is a dimension bar of about 1 millimeter. So these are about 0.5 millimeter diameter. And we were able to reconstruct them. The next step was we took an earthworm, simple object to test on, doesn't need as much approvals from the university, it turns out. It doesn't have, if you don't have a spine, it turns out, there are much fewer rules. But you can see that the vessels inside an earthworm we're able to detect them, and also the outer layer. So we're getting, again, some indications that the resolution potential is also there. We're getting closer and closer. The one interesting question that came up is the age-old problem of polarization. If you look at the body, all the blood vessels are oriented in specific ways. If you go in the E field that's orthogonal, you will not be able to see it. So polarization becomes important. If you've designed radar, you know that, but this is a different problem here. So we looked at capillaries again. These are very small capillaries, and we saw if you apply a simple unipolar, sort of a unidirectional polarization, you won't be able to see orthogonal vessels. But if you apply the right polarization, orthogonal, if you do with the circular polarization, you can actually see both blood vessels. Right? So that's one of the things you have to take into account when you're designing an E-field excitation. Now, the next step I want to go into is, well, what I've discussed so far is a system that's essentially a contact-based imaging system. It's low cost. It could be widespread. It has the potential. I'm not saying we've solved the problems. We're heading slowly towards some goals and milestones. It's a long project. But the next step we were interested in is, can you truly make it non-contact? And the benefits of non-contact are numerous. If you have a non-contact system, by definition, it's not operator dependent. So we can talk about machine learning on ultrasonic imaging and all these things all we want, but if the operator needs to be there to kind of move it around up and down, that is limited in terms of how much you can actually do it by machine and automate it, right? So our vision was, can you make it non-contact so you remove the dependency to the operator, and at the same time you might have applications where you can't actually touch the surface of tissue, or you don't want to touch the surface of tissue. There might be medical or non-medical applications. Okay, so in that realm, what the imaging system would look like is you have ultrasonic arrays interleaved with RF arrays. So this is going RF in, ultrasound again. The big difference is you have a multi-layer system, but now you have air between this layer, the final layer, which would be a tissue, and your detection. So the question is, can you do that? And the big problem here is, if you've paid attention to the acoustic impedance of different targets, you see that tissue, if you look at this is tissue, and this is my object, and signal's coming out, tissue has an acoustic impedance is about 1.5 megareal. Air, on the other hand, has an acoustic impedance of 400 rail. So you can imagine propagating from tissue outside, there's a huge impedance mismatch. So what happens is you lose 65 dB of signal in pressure going from tissue to air. This is why when you go to someone who does ultrasonics and say, oh, I want to do ultrasonics in air, they'll start walking away, most likely, because they understand the 65 dB problem. Now, there's a little bit of silver lining here, because when you go in air, the impedance is lower, so the mechanical noise floor is lower, so your sensitivity is better. So what you really lose is about 30 dB. This is a huge number, but like transducers or CMOS. These are detectors that are capacitive in nature. If you imagine a capacitor that has a very large electric field, is biased, and vibrations will give you the VDCDT sort of thing, and you get current injection. So it's a MEMS device, picks up vibrations. And we need to use something like this, because if we are overcoming 30 dB of loss, we need to use ultra-sensitive transducers, and we need to have large arrays, because we need all the gain that's possible. We need to do SNR averaging in space and time to get the signal back. So the nice thing about CMOS is that I can have wafer-level solutions in large arrays of these devices operating in air. Right? So that's where the technology comes in. Again, where does the technology come in? Well, this is where one of the technology pieces is a very important piece of the puzzle. Now, when you're looking at this imaging problem, there's another layer of complication, and that's when you go from the source, which is your tissue, to air. Yes, there's 65 dB reflection is a huge problem, but there's another problem. The speed of sound is different by factor 5. You get huge refraction. So it's like looking from inside water outside. Everything is going to get refracted. So here I've amplified the signal in air by a factor of 5,000 to be visible. And this is the sort of thing I'm getting in my angle. You can see there's a huge refraction. What I'm measuring here is very different than what's going on here. So if I have the field measured out there, the reconstruction to inside is not trivial. I can't apply my simple radar equations, SAR equations to this problem because the speed of sound is so different. So I'll give you an example. This is our optimal reconstruction technique, which we do a layered SAR technique. And we're now moving to techniques that use the Green's function for the problem and apply it to get more focused response and more corrections for the edge refractions from multiple layers. But if you don't, you see that you see actually target in a different location, you completely lose your signal to noise ratio and resolution. Right? So that's a problem with just the speed of sound and refraction. So this video is basically showing our system. So this is the receiver. Right now it's only 4 elements. We're using 1 element at a time, so we're not doing the array. And the electronics is on this board. There's a target that's sitting inside this agar sample. Agar is replicating tissue. And as I move this to synthesize my array, I can see in real time the image reconstruction. And more points combined, I get more and more resolution. And I'm able to reconstruct the object location. So this is not yet using the full array. This is not using wafer level arrays as we hope to use. But it's showing that with this sort of system, I'm able to reconstruct the object at a given resolution. And we've looked at single objects, double targets that we're able to reconstruct, again, moving very slowly towards showing that more and more complex targets are doable. This is a completely new area, so whatever problem we face, we can't look up the answer anyway. It's not like, oh, we go and look up non-contact imaging, there's a book on this thing, and we open it up. You have to sort of figure out the problems as you go. Another advantage that we get, one probably small advantage that we get, is that when you come from a high speed of sound to low speed of sound material, you sort of refocus your field. So what the material is essentially giving you is it gives you this large of an aperture effectively for a much smaller aperture because it refracts the rays into your system. You can think about this as like having a lens. It's like I have an automatic lens sitting on my target. All the beams are focused into my transducer. So with doing this, I can improve my resolution. So what we've shown here is that your homogeneous material has this sort of range resolution with aperture. But if you do layered, you get much better resolution. We're able to, even with single frequencies, able to reconstruct and achieve something like 3 centimeters resolution. If we did not have this phenomenon, you'll get something like 24 centimeter of resolution. But by having this phenomenon of focusing, we can get a little bit of resolution back. So this is one small silver lining with all the refraction going on. Here I'm showing multi-frequency, so our goal is to actually do multi-frequency SFCW type of imaging. And on the left, I'm showing single frequency images from 9 different frequency points. On the right, I'm showing the reconstruction of these. As you combine different frequencies, your images improve. And on the rightmost figure, I'm showing you the actual image with gamma correction. So we get once again, we get closer and closer to these targets. There are still artifacts, and we're trying to figure out how to suppress these artifacts in the next steps. We also did some tests external, so our funding agency wanted us to send our devices to an external lab so they can independently verify that these things work because they wanted to look at it from a large array of targets, different size, different shapes, different materials. And this is what we have based on their data. This is the ROC curve. This is probability of detection or true positive rate. This is a false positive rate. And we see that with not any post-processing, so we're not applying here any machine learning, classification, nothing at all. We're sort of looking at more or less raw data, just removing the baselines. We're able to get about 90%, 15% detection false alarm rate. And this is, again, their data, not ours. Okay, so let me change gears a bit. So we talked about imaging systems that are sitting around the body. We've moved away from non-contact imaging. What are some other platforms that we're working on? So in another platform that we're working on is we're looking at what about inside the body when we do have access to. So the parts of the body that we have relatively good access to, an important part of that is the GI tract. You can have a pill, you have a GI in your GI tract, you can monitor the GI tract. And today there are current systems. There's a pill cam by Given Imaging. It's growing at a very fast rate, and it does a lot of things, and the most important part is that it basically takes an image of the GI tract. So if you have internal bleeding, doctors actually use this today to figure out where the location of the situation is. So this is what's being used in the hospital today. Lots of new applications of this. So we started looking at this problem from a slightly different perspective, and this is in collaboration with Professor Kuryakov, which is looking at all the ultrasonic devices and CMOTs and arrays and flexible arrays. Also looking at working with two professors in the Stanford radiology, Eric Olcott and Brooke Jeffrey. And the idea is we already see that ultrasonic systems are going from large devices with help of electronics, more advanced electronics, things that in this community they're designing, probably a lot of TI chips in these modules. They're going to smaller form factors, and we asked sort of the crazy question to see, can you go one step further and put it on a pill? So you have a capsule that has an ultrasonic imager sitting on top, has an antenna that sends out the data. So you have the capsule as a small battery inside. It's taking images for 4, 5, 6, 8 hours, and it's transmitting these ultrasound images outside. Right? So that's what we're going after. And the system would have an RF transmitter. It will have an ultrasound array and electronics. Everything is integrated into a small capsule. That's sort of the application we're going after. And once you have the system, you can actually get images of the internal structures of the GI tract. Now, there are lots and lots of problems with this thing. One question is, well, if I don't know where my imager is, how am I going to interpret the images? We don't know. We're looking at that problem because the imager is tumbling and turning, and the doctor's not going to be able to orient these things automatically. So some machine learning tool has to come in and figure out how do you orient this and put it together. Right? Second problem is, well, how do I figure out if I see an anomaly, how do I go back to it? How do I focus into it? How do I image it? If it's all going through, it's kind of a one-way system, can I go back and figure out more images? Well, you need an adaptive system. You need sort of on-board processing, and that's one of the things we're looking at. So there's lots and lots of questions here. Well, essentially, the platform we're pitching is a disposable and wireless capsule ultrasound. And so you have a system of ultrasound. The cost is so low, you can dispose of it. And this is one of the things, the notions that, again, in this community we like, because we would like our electronics to be thrown away. If you could make us throw away our mobile phones in a month, you would do that as opposed to 2 years. If it's a day, you would do that. So this is one area that you would have to sort of throw away the silicon on every use. And this is kind of showing the GI tract. So what is the system? The first system we're designing is actually looking 3 parts. We designed the array, the front-end electronics, the RF transmitter. This is what the form factor looks like. This is kind of our first prototype mock-up of the system, which has the antenna and the ultrasound array. We're now in the process of characterizing these elements, the ultrasonic elements. We're looking at the transmitter. We've done some tests on the transmitter. And also this is the 128 elements array that we're testing in the lab. And these are the students from Professor Kuryakov and my lab working on this problem. The transmitter side, to just tell you a little bit about the transmitter. So the transmitter, the problem is you have to go through a lot of tissue. So the pill can go to as much as 15 to 20 centimeters of tissue you have to come out. So designing a transmitter for robustness, you have to be careful about the link budget. We're using about 400 megahertz. An FSK system has 2 PLLs on the top and bottom, and they're ping-ponging based on the bit sequence. So both PLLs are ultra-low power, operating simultaneously. So I'm not doing a fast locking PLL into power consumption. Designing 2 low power PLLs, and I'm ping-ponging between the 2 sort of for FSK detection. And we've done some tissue phantom studies to about 10, 15 centimeters. Up to 10 centimeters, we're comfortable with 10 to the minus 5 bit error rate. After that, it depends a lot on what the material is. So we're looking at better phantoms today and figuring out what our limitations are. The bit error rate is sort of capped at about 10 megabit per second. There's an optimal point in terms of bit error rate, signal to noise ratio, and your link budget and frequency. So those are the 2 topics. Now, in the last part of my talk, I'll get to getting inside the body now. So we've talked about outside the body. We went a little bit inside the body. We kind of toyed with GI tract and pills and capsules. But the last part, we want to just get the gloves come off and we go inside. We talk about implantable devices. Now, the big area that we're interested in, the reason we talk about implantable devices is that there is an emerging field of bioelectronic medicine or electroceuticals. And the idea here is, and there's been a lot of studies in this, very interesting results, is that you can tap into the nervous system and cure specific disease. A lot of the organ function is maintained in different disease structures. What you need to do is you need to understand how to tap into the nervous system and take control of that organ. So if you're a diabetic patient, insulin release could be controlled theoretically by the nerves that go into the pancreas. So if I can map the nerves go into the pancreas, I can control insulin release. I don't need to have an implant. I can also measure a lot of parameters by tapping into the nervous system because the body measures them on their own. So I need to figure out, decode what the body is doing. So that's the idea. And the basics of it, the reason that this community should hopefully be excited, and I think they are, and we had an SRC workshop a couple of weeks ago looking at this problem, is that electronics will be replacing drugs in a lot of disease, not everything, but in a lot of disease. And the pharmaceutical industry is huge, a lot of problems. And attacking it from an electronic point of view could have major advantages. And one of the advantages is that you don't have generic solutions anymore. I don't go one size fits all, I get a pill, and everyone my weight gets the same pill and I have it. They will be fine-tuned to your parameters. You can have closed-loop therapy that's fine-tuned to your state. And more importantly, you don't get systemic side effects. When I have chemical drugs, there's systemic side effects. Everywhere in the body gets the target. Here, again, designed right, I can potentially get into the target of interest. Lots of disease being looked at today, immune system being one major part of the study. Asthma, arthritis, hypertension, diabetes. So people are not just looking at neurological disease. This is the point I wanted to make. I didn't even point to neurological disease. I know that the nervous system, the central nervous system, you go in, you can do DBS, deep brain stimulators for Parkinson's and so forth. This is the direct electrical to electrical. Here we're talking about autonomic system. We're talking about other organs. We're talking about taking control for hypertension, for different disease, not just the central nervous system. So what does the system look like from an engineer's point of view? The engineer's point of view, you have three parts. You have the recording, diagnostics, and treatment. So you record the parameter. So let's say that recording could be a glucose or insulin. You measure that in vivo. Then you run some kind of a parameter analysis which is tuned to you based on your baseline, figures out, well, your insulin level should be this time of day based on what you had. And then you go in and you tap into the nervous system and you, in a closed loop fashion, control the release or lack of release or prohibition of release. So you need all three parts to do closed loop. So if you think about it from electronics, IC design, system design point of view, I need a sense, a force, some kind of computation, which may or may not be local. You don't need to have all the process in the implant, but you may need some for security, for latency, for energy. And there's duplex communication. You also need these to be super small and operate at depth because you don't want to go in necessarily with something that's large if you want to treat a disease that is simple. If you want this to be widespread, you need devices to be minimally invasive, so surgically inserted as opposed to open surgery. And you need to have better biocompatibility. So again, electronically, you need reliable duplex comm. You need power delivery because I don't have a battery in there. It's a big reliability headache, and I don't have enough power. And I need specific targeting. So you look at that and say, well, why hasn't this picked up as much as it should? Because the fundamentals of these things have been understood for a while now. And the problem is these are the type of electronics we're thinking about in the body today. They're not the type of thing you want to do if you have hypertension. I don't necessarily want to have a brain surgery and put these in if my blood pressure is a little bit up. So today's implantable devices are essentially last resort. You need to be sure that there's no other solution, and then you go and dig in with the electronics. And you can see the devices and the solutions today are very large, very bulky, and not the type of thing you can imagine for widespread use in a larger disease states. Oops. So the problem that you face almost as soon as you start looking at this problem is how do I get power transfer to the system? And it can do power transfer multiple ways. The one we are familiar with, all of us, is inductive power transfer. So you have two coils or multiple coils, and you do power transfer from one side to the other. This is actually older than wireless communication. Wireless power goes to Tesla in 1880s. Wireless communication was a little bit later than that, so it's been looked at inside out. What you really, the three parameters are coupling constant, the coefficient, the Q factors, and the matching. And the problem with any sort of coil power transfer, if you've designed one, is that it's very difficult to get very high Qs. So if I look at this equation, I really need high Qs and high Ks. At least one of them needs to be very high. Difficult to get high Qs, the frequency of interest. So that's one problem. The other problem is the coupling coefficient goes as 1 over d cubed. So if you try to get inside the body, and especially if your range to size ratio, this is our figure of merit, the range to size ratio, if your range to size ratio is large, then your coupling coefficient is going to be very small. You're not going to be efficient. You're going to be sending power to everything but the receiver. And the third problem is the near field operation, the transmit and receive are codependent. If I want to take this circuit, and if I ask you, well, please design the best transmitter you can, you have to ask me, well, what does the receiver look like? And where does it sit? Because they're dependent, they're not independent. The next solution, potential solution, which is, I think, a lot more interesting, a lot more potential, is that you can look at getting and using propagative modes or radiative modes into tissue. So you can say, well, why do I operate purely in the near field? Can I increase the frequency so that I have propagating modes into tissue, right? Just wireless communication. I have propagating modes, I have radiative modes. And this is very interesting for a lot of applications. The limitation is, if you again design antennas, you know that once the wavelength is much larger than your implant, so the implant here is about 1 millimeter, your wavelength, even if you go to gigahertz frequencies, is in the order of centimeters or larger, 5, 10 centimeters, depending on what your frequency you're using. If the size of your antenna compared to the frequency is much smaller, you're going to be inefficient on both sides. You're inefficient in receiving the power. If you've ever designed a CDMA antenna or a wireless antenna, you'll see this. It's going to be inefficient on the receive side, and it's also going to be inefficient on the transmit side because I can't focus the energy in. In order to focus, I need a small wavelength. If the wavelength's large, I can't focus it in. So what you usually see are systems that look like this, which the energy is deposited in larger sort of volume structures as opposed to focusing in. So I get 1%, 0.1%, 0.01% efficiency. The rest of the power ends up other parts of the tissue. This is great if you have one acute setting, but over time, chronic applications, this is going to be a problem. And the other problem with it is, if you look at this focusing effect, ideally, if I don't have any tissue losses or tissue losses are small, I can focus the energy in. But as the tissue losses start getting to realistic parameters for body tissue, you see that the focusing gain also drops because the losses at these frequencies start dominating. And so we know that. We put stuff in the microwave oven, they heat up. So we know that at 2.4 gigahertz, something must be absorbing. So the question is, can you optimize? And for some applications, you can optimize. For some others, it becomes problematic. So when we started looking at this, we said, okay, the big problem is I need to have a small wavelength. And where do I get a small wavelength from? Potentially, I can get it from ultrasound because the speed of sound is 5 orders of magnitude slower than the speed of light. So I can get 5 orders of magnitude smaller wavelength. So if I want a 1.5 millimeter wavelength, all I need to do is operate at the megahertz in ultrasound, and I get my wavelength. And megahertz ultrasound, we know how to do. It's not sophisticated. We've done it with ultrasound imaging. We've even gone higher in ultrasound imaging. So once you have this, you have better focusing. You have better receiving or reception of energy. And you can also have network of implants. You don't have to have one. You can have a network of these devices that you sequentially power and communicate with, either TDMA, FDMA, or CDMA. You can communicate with multiple devices at once, or SDMA, which is space division, multiple access. So focusing in tissue of ultrasound, which is a big problem, has already been done for other applications. People go for ultrasound imaging, for HIFU, this is high intensity focused ultrasound. They've already figured out how to focus the energy in. You also get side benefits, and the side benefits are that you have larger FDA intensity and lower tissue losses. These are the side benefits you get with ultrasound. So in the past 5 years, we've been looking at a lot of projects figuring out what is going on in ultrasound powering these devices. So we started out by investigating just the basics of ultrasound power delivery. Can you get enough power in ultrasound? Can you design transducers that do power transfer? What are the impedance levels? How do you model them? There was a lot of modeling, the mechanical structural modeling of the transceivers, of the receivers, more or less. So how do you optimize the modes that you're interested in? What type of modes do you want to operate? How do you recover the power more efficiently? Then we started building these implantable devices. So the first one I'm going to talk about a little bit today was combination of ultrasound and RF and had some delivery for power. We also did some localization and beamforming. Can you find the implants and focus power into them? Optogenetic, can you apply a photonic excitation? Can you do an optical excitation in the implant? Drug delivery, we're working with Professor Zaire in the chemistry department. We've shown that we can use electroresponsive polymers, have these implants in there that are loaded with the drugs. When you power them up, they apply electric field, they release the drug. So you can actually do traditional drug delivery systems. More recently, we looked at duplex ultrasound links. So now we can communicate back and forth with the device ultrasonically. So data and power go with ultrasound and comes back with ultrasound. We are going to present a pressure sensor in VLSI, which is sending variable ultrasound and minimization. So let me quickly go over a few of these examples in the last 5 minutes or so. So the major recipe in the system, when you're designing this ultrasonic system, is that you need a receiver. So just like antennas, you need to receive this energy and translate it to DC. So what we have here is the receiver is a function of multiple parameters. It's a function of your material, dimension, packaging, acoustic loading, and also electrical interface. When you're done with the image, with the modeling of this thing, what you'll end up having is an impedance profile that looks like this. You have a series resonance, you have a shunt resonance, you have some kind of radiation in each of these, and you have an inductive band in between. But more importantly, perhaps, you have other modes, undesirable modes that kick in. So the name of the game here is how do I optimize the desired mode and suppress the energy that's coupled in undesirable modes that can come in and reduce my overall efficiency. So that's what we're designing when we're designing transducers. So we have an array of these transducers so far designed. We have designed in PZT4, PZT5, and barium titanate. The way we define efficiency is aperture efficiency. If you design antennas, you're familiar with aperture efficiency. This is essentially saying that I take my actual receive effective aperture and divided my physical aperture. That gives me a ratio. That is what I call my aperture efficiency. In other words, what is the electrical power that you've recovered? Divide that by the acoustic intensity multiplied by the actual physical area. So that is my efficiency. If I get 100%, that means that all of the energy that came in that acoustic intensity, I can recover, right? So what you'll notice, which is interesting, is at some point your efficiency can actually go above 100%. And this could be exciting because why does efficiency go above 100%? Now, if you design antennas again, this is familiar because antennas that are small, they have an aperture that's larger than their physical area. If you think about a dipole, it doesn't have any area, but it still picks up energy, right? So what this is essentially saying is in small antenna, quote-unquote, systems, you can get more than 100%, just meaning that your physical aperture is smaller than your actual aperture that's picking up energy. Sub-wavelength resonance, this actually could happen, happens in antennas all the time. So don't get surprised that our efficiency is going above 100%. So this blue here is showing the impedance profile. The red is showing the efficiencies in 3 different sort of material systems. Measured efficiencies, 3 different material systems. Now, the first implant we designed, we said, okay, why don't we receive the power and data in ultrasound, and we communicate back data with RF. So we're doing a hybrid system. Data comes in ultrasound, goes back in RF, and we also have a delivery system. So we have 100 microamps at a volt that it can do whatever you like with. This gives you available power in your platform to do stimulation, sensing, if you want to put a glucose monitoring system, whatever you want to put, this allows for that. What does the transmitter look like? The transmitter was a PPM system, 2 nanosecond pulses, operating at 4 gigahertz. So the transmitter is a pulse-based, pulse PPM style modulation system at 4 gigahertz UWB, and the receiver is at the megahertz. This is what the chip looks like. Not a lot you can see here except for the RF link. The inductors always show up, so that's interesting. This is what this is our first sort of packaged implant. It was interesting looking back at this. So 4 millimeters by 7.8 millimeter. You have an antenna that's sitting on the edge that's doing the wireless uplink. And you might ask, why did you use RF for uplink? Didn't you just say RF is bad? But yes, it's just that the link budget in uplink is very different than downlink. In downlink, you want to power it up, so you want minus 20 dBm, minus 15 dBm to get there. In uplink, it's just data. I can do minus 80 dBm, minus 75 dBm. right? So it's a very asymmetric link when you think about implantable systems. We did some blind tests at about 3 centimeters of tissue phantom. This is the SNR we're getting, which is sort of commensurate with if you wanted to do a ppm modulation. The next example is we wanted to see if we could do optogenetics. And the idea here is you have light sources that are implanted in, you genetically program tissue to be responsive to light, neurons to be responsive to light. This was incidentally invented at Stanford. And you basically can stimulate the neurons selectively based on the wavelet you use and the optical stimulation. And the problem is, for small animals, this is okay because you can go with a fiber and the brain is so small you can get in there. For larger animals, it becomes a problem. If you want to do this in primates, you're going through centimeters of tissue, and it's a much bigger problem to worry about. And you need the high power levels because now you're optically stimulating the system. So I won't go into the details of this, but what will happen is the aperture efficiency remains the same, except that if you look at the conceptual system block diagram, I have 3 parts. I have the receiving acoustic energy, I have a rectification part, and I have the optical stimulation. These are highly nonlinear, as you can imagine. So the efficiency is going to be function of your power level. Just like any rectification system, it's nonlinear. The impedance levels are nonlinear. So the name of the game is to be able to optimize. You have 4 different stages. You have acoustic, electrical AC, electrical DC, and optical. And the name of the game is to optimize the chain so you get the maximum acoustic to optical efficiency. Right? So the chip is optimized to give you the maximized acoustic to electrical optimization. Let me go through this a little bit quickly. So how do you come up with the optimal point? You have models that look at different power levels. So every power level will have an optimal impedance. Because it's a nonlinear system, you run this iteratively and you find the optimal piezo impedance based on a power level. Now the question here is, for a given power level, the optimal impedance is fixed. The way we get around this, I'm not going to talk about here, is you can actually put a closed-loop system around it that adjusts the frequency in an ideal way, so you get the optimal impedance for every power level. Right? So that's something you could do in a closed-loop system. We have a paper on that in case you're interested. The other interesting part is that the impedance is also a function of your aspect ratio. So that another parameter to optimize is your aspect ratio of your transducer determines your sort of impedance levels. Finally, these are some of the measurements. You can see here's the acoustic intensity. This is the FDA limit on the right. And on the left is the optical intensity. So for example, if you're looking for 6 milliwatts per millimeter squared of intensity in optical, you can do that with about milliwatt and a half ultrasound. Now these are for different areas. That's why the numbers on the x-axis and y-axis are smaller. I'm not getting more than 100%. Is that the optical transducer or the optical LED is much smaller than the acoustic. So the kind of patterns we need here are about 1 to 10 hertz. So we're going up to about 50 hertz and about 50 milliwatts per millimeter squared, which we can actually hit with some levels of acoustic intensity that's lower than the FDA. So this is a video. I don't know if we can play the video. Let's try to see if this works. No, that did not work. The newer version of the implant, we have a 3D printed shell. But the video I did want to show, if possible, is this one. So if you just, okay, thank you, thanks. So this is the implant sitting in. This is a sill guard sample that's replicating the tissue. And you can see that going through the depth, we can power them up, and the LED is sort of lighting the sample. So thank you, thanks for. So you can go through the sample, and it can light up the implantable devices. Everything is powered and communicating with ultrasound. So after that, we have a new chip that we presented at ISSC last year. And the idea was now you can go in with ultrasound and come out with ultrasound. So you get rid of all RF systems. And so you're going with 1 megahertz and 2.5, so an FDD sort of a system. And we are getting about 25 kilobit per second data rate in receive and about 100 kilobit per second data rate on transmit. On a 10-bit PRBS, we're getting about 10 to the minus 4-bit error rate. So this is sort of our wireless system ranging capability. This is what the packaged implants look like for this communication system. We're using now polyimide substrates. We cover it with an epoxy. We're moving towards medical grade epoxies. We're sending some of these devices to the FDA. They're running their independent tests to see what we're saying actually makes sense. They're doing thermal cycling, looking at long-term stability and a lot of other questions that are being answered. On the right is what the chip looks like. This is a test in a tissue phantom. Again, we're going 6 centimeters in. Now we're getting to a range of about 6 to 8 to 10 centimeters. Most of the applications we're interested in require robust links to about 6. Some applications like bladder control wants you to go about 10 centimeters in. So this is what we started looking at, 6 centimeters and above. And you can see the 2.45 gigahertz, the 2.45 megahertz output spectrum there. The next implant is we did a two-mode implant, hybrid implant, does optical stimulation and electrical stimulation. So here we're showing the stimulation pattern. This is fully biocompatible, barium titanate, and we have 6 centimeters in porcine and 9 centimeters in a tissue phantom. And so one of the things we wanted to do is we wanted to see if these stimulation patterns are actually working on actual in vivo samples. The problem is when you take a frog or a rodent, the animal is so small, you're not going through any tissue, so it's relatively easy to do this. So we put together a setup that combines the two hearts of the problem. So we have the biology sitting on the right, so the in vivo sample on the right, but on the left we have the phantom to show that we can actually wirelessly power it. So we wirelessly power the implant, take the wires out, and stimulate the nerves on the right. So what this video is supposed to show, if you can, I promise this is the last video, I didn't realize. This is more interesting than the other one, depending on your interest, I guess. So this is stimulation of a frog leg, and we're powering the implant, putting different sequences in, and you can see that we're going to start with a 2 hertz stimulation, and then we're going to change the stimulation patterns for half hertz and up to 5 hertz, and we have responses on the tissue. So let me skip it. Finally, we have a pressure sensor, again, bidirectional ultrasound. We're measuring pressure on a transducer, on a piezoresistive transducer, 10-bit A to Z, and then send data out. These are the measured parameters of the pressure. We can get about half a millimeter mercury pressure difference on a range that's from 400 to 1200 millimeter mercury. So this is for a full bladder on top of Everest, all the way to an empty bladder on seafloor, right, or sea level. So that is, we're hoping that we don't basically get out of this level in terms of pressure. So in conclusions, brings me to my last slide. I hope that I made a case that there are some problems in healthcare, whether it's accuracy or efficacy or cost. And one big problem from our point of view, there's a Nyquist problem. We're not sampling our health state or disease state frequently enough. And I hope that I was able to introduce a few new medical technologies, or at least the path towards medical technologies, I should say, that could potentially address some of these problems, whether it's more frequent testing, getting to personalized care where you match the treatment to disease to the patient, or for something like a sort of a disposable monitoring system where you're monitoring and it's so low, the system is cheap and low cost, you can do it at a much higher frequency. And last but not least, this is my group. These are the students who actually do all the work. I would like to thank the funding agencies and our collaborators at Stanford and outside. Thank you. Any questions, I guess? Yes? Yes, so it depends. In some ways, so the question was, can we use machine learning algorithms to sort of overcome accuracy of sensors? And sort of the answer to that is it depends. How is the sensor deficient in terms of accuracy? Is it fundamentally deficient that the data isn't there? Machine learning can't help you if there's no information coming out. But if the information is noisy, the information is noisy, or there are patterns that you need to extract, then it comes and helps you. So you can't have, it's an information theoretical argument that if the data that you're capturing from your sensor does not exist, the information does not exist, machine learning can't extract anything. But if it exists and it's distorted by noise or other parameters, then it can help you quite a bit. So I guess it depends. I think the answer is depends. Yes?
So, yes, it was a great presentation.
Thank you.
Sorry. You showed some ways to do some reconstructions, and I was wondering if you just give a brief overview of whether you use like a full wave solver or a geometric reconstruction algorithm or what.
So the reconstruction imaging.
Yeah, in the imaging, and you showed multiple imaging types. So I guess just your favorite.
Yes. So
I definitely like the standoff.
Yeah. So our bread and butter is always a form of SAR. So now that here you get into trouble because you call it one thing and others call it different things. So I'm just going to call it frequency domain SAR. That's the one we predominantly use for most of our applications. What we have worked on time reversal techniques. And what time reversal does is that you sort of backpropagate the field to a given interface, and then you can do another algorithm now. So we're looking at, for example, backpropagating to the surface and then doing a SAR going back. So those are the things we've looked at. In addition to frequency domain SAR and sort of backpropagation, we've done some forward solving techniques. And the idea of forward solving is let's imagine the target was point X, let's capture all the fields in a forward solution and put it in next position and next position. So I have a solution for all the target locations, and I put it in a feedback loop and figure out where I am. And that's computationally very exhaustive, and it's not fast, but it's most accurate if it converges. So the question about iterative methods or does it converge, we have some ideas. We published a paper maybe a year ago that we looked at specific filters that make the convergence a little bit more certain, let's say, and make the forward solver a little bit more mainstream or something that we could use. But the trade-off between speed and accuracy always exists. Yes, predominantly frequency domain SAR techniques. We might call back projection techniques, yeah.
Oh, that's great. I was wondering, you just mentioned speed. What kind of speeds were you getting? I noticed when you showed reconstructions, when it was scanning across, it looked like it was in real time. Was that
that's real time because we're not doing very complex. So this is a layered SAR reconstruction. So whenever you so the beauty of frequency domain SAR is that if I do a time domain SAR, I need all these filters at different points. Frequency domain SAR is nothing but an FFT. So we make it sound fancy and we say frequency domain SAR, but we're running FFTs. And so if you do layered SAR, you run FFTs plus boundary conditions. So it is as fast as you see. It's even perhaps faster, but it's not as accurate. So you see there was ghosts on the targets and things like that. That's the problem.
Yeah. Great, thanks.
Well, thank you, thanks.
Thanks for talk. I have a question on the assembly part, maybe. So you mentioned you have the acoustic piece, which is a PZT. How difficult is like in machining this piece and assemble to an IC with some non-conventional material that we will deal with in the IC process? Is that still in a tricky lab manual process, or there's a way to make it easier for batch fabrication, hence for low cost? Thank you.
So that's a good question. So I think the quick answer is it is a big problem and it's still not in batch manufacturing state. So whenever our collaborators, for example, FDA guys say, send us 10 samples, that just means like the students are going to spend more time in the lab. We don't have that capability. But nevertheless, the reason we're using polyimide substrates, flexible substrates, is that we want to use materials and components that could potentially get into the batch manufacturing state. So that's what we're working on. But there are a lot of unknowns. I don't want to say, when I came to this field originally, I came from a millimeter wave design field. So everything was 100 gigahertz, 50 gigahertz. And I thought, a megahertz will be okay. We don't have to worry about parasitics. Little that I know that the wavelength is still very small. So the wavelength being a millimeter, that means that every 100 microns of trace or packaging or your backing will be a tenth of a wavelength, just like it is at 100 gigahertz. It just happens to be at lower frequency. So the hardest problem of all, I think, is the mechanical questions you need to answer. So mechanically, can you make sure the right impedance is presented? Do you have it broadband? Do you get it efficient? So for example, when you put the PAs on a board, that's the first thing, most of the waves are going to get launched in Rayleigh waves or Lamb waves into the structure. So you get surface waves, all the energy actually is exhausted in the substrate. Now you need to look at structures that actually filter out that effect and get the wave actually in the desired mode. One thing with acoustics versus RF is that there are many, many modes. Any material that sits on the other material, there are many modes that can actually sit between these two. So the question is, can you efficiently couple the right mode and extract that energy out. So it's not still, the quick answer is it's not batch mode. We have some way to go there, but we have the recipe or the fundamentals to get to a batch mode. For example, that's the path. Thank you. Next. Yes.
Thanks, Professor, for the very interesting talk. I have a question on the ultrasound power charging.
Yes.
So how do you need the externally design a different antenna, and then also how far away can you do it? Like what's the distance? Related to the propagation properties, right, of ultrasound.
Yeah, so the propagation properties in tissue don't change as much, except when you hit bone and air. So if you want to go through air in tissue, just like we saw the 65D, you don't want to power anything in the lungs. That's a very difficult thing to do with ultrasound because you have to go through the impedance difference between air and tissue. The problem with bone is a problem that we're thinking about, but we are looking at shadowing, similar to a shadowing problem we see in RF. And shadowing, we have techniques to get around to some extent in RF systems. So to answer your question, I think most of the tissue structures we're looking at so far are relatively homogeneous, even though their mechanical properties change, but that's plus minus 10%. If you go from fat to tissue, the mechanical property changes, variations are small, unless you hit bone and air. And when you go through bone and air, for example, go through the skull is a challenge. So we are actively thinking about ideas there. If you have some good ideas, let us know. We're thinking.
Okay, thank you very much.
Thank you.
Let's wrap up. We'd like to thank our speaker for giving this very exciting talk.
Oh, thank you. Thanks
by a certificate of appreciation.
Thanks very much.
Signed by both SSCS chapter chairs and also EMBS, our co-sponsor. John Black, the chair of EMBS, he couldn't make it tonight, but we present it to you.
Thank you. Appreciate it.
Okay, so we'd like this. Okay, thank you very much all for attending this seminar tonight. Please check out our website for our upcoming talk seminars. We're going to have a large conference on intelligent systems and machine learning. It's coming up in early November. Please stay tuned and also subscribe to our email list. It's the instructions. If you got the email, the instructions is at the bottom of the email. So then you would be updated with all the seminars that we're going to have in the future. Thank you very much. So let me turn it.